This is the lecture for Chapter 40, Digestion and Absorption for Anatomy and Physiology 250. All right, we'll try to get through. Let's see, this is about 45 slides. We'll try to get through it pretty quickly, though. I will tell you what parts need to be emphasized. Now, here are some of the learning objectives. We're going to describe the primary mechanisms of the digestive system, define mechanical digestion, discuss mastication, explain the three main steps, stages of deglutition, differentiate between peristalsis and segmentation. And we're going to explain the process of emptying the stomach. So here's an overview. Primary function of digestive system: to bring essential nutrients into the internal environment so that they are available to each cell of the body. So the digestive system mainly is there to bring essential nutrients to the internal environment. Remember, nothing comes into your body until it is absorbed through the intestinal wall and gets into the bloodstream. Then it's considered inside the body. Other than that, the alimentary canal or digestive tract is considered to be hollow tube that runs from your mouth all the way to the anus, the hollow tube that's outside of your body. So the mechanisms that were used to accomplish the primary function, which is bring essential nutrients to the internal environment, is we use ingestion and digestion. Motility of the gastrointestinal wall uses secretion, absorption, elimination, and regulation. That's the mechanism of digestion. I'm sorry. Now we have mechanical digestion, which changes the ingested food from large particles into smaller particles or minute particles that facilitates the chemical digestion. So facilitate means to help. So mechanical digestion breaks down larger to smaller to help out with chemical digestion. So mechanical digestion also churns the contents of the gastrointestinal lumen. Lumen is the empty space. So the empty space inside the gastrointestinal tract to mix with digestive juices and ensure the contact with the surface of the intestinal mucosa, which helps absorption. Mechanical digestion also propels food along the alimentary tract, eliminating digestive waste from the body. Mechanical digestion also includes mastication. Mastication means to chew. Then you have deglutition. Deglutition has three different phases or stages. One is the oral stage. So that's from the mouth to the oral pharynx. Oral pharynx is at the back of the throat. That's voluntarily controlled. When it's voluntarily controlled, you know that the muscles that actually help with deglutition are skeletal muscles. Then you have a pharyngeal stage that goes from the oral pharynx, which is the back of the throat, to the esophagus. In this stage, you have involuntary movement. So it's a combination of contractions and gravity that moves a bolus into the esophagus. The bolus is the food that has been chewed and taken to a ball, and then when it's swallowed, it's considered a bolus. Then you have the esophageal stage that goes from the esophagus to the stomach. It's completely involuntary. Here's some of the stages. This is the mouth. This is the bolus. So when it's ready to be swallowed, this up here is the hard palate, and this muscle is the soft palate. And this little part of the soft palate sticks down is the uvula. So this part at the back of the throat is the oral pharynx. This first oral stage is all voluntary. It's all skeletal muscle. And so it's going down further. Tongue pushes up against the hard palate, pushes the bolus down further into the oral pharynx. This is all voluntary. Then you have the pharyngeal stage, where it goes down from the pharynx, which is the oral pharynx, laryngeal pharynx, and it goes down into the esophagus. And so if you notice, this is the trachea at the anterior portion in the esophagus is posterior to the trachea. So as the bolus comes down, it hits this little flap. This flap is called the epiglottis. And so that's this flap here. It's the epiglottis. The epiglottis typically is open when you're not swallowing, so it allows you to breathe consistently. And so when the bolus comes down, it pushes the epiglottis down, closes off the trachea, and allows you to swallow the food. And that's what it's doing here. Pushing the epiglottis down, it will uh, it will close off the trachea, and it will allow the esophagus to stay open, and that's where the food is pushed down into the esophagus. Now, this pharyngeal stage is involuntary. So the muscles that contract and actually pushing this along are involuntary. Then you have the esophageal stage. It goes from the oral stage to the pharyngeal stage. Then it goes down to the esophageal stage. This is where the epiglottis is closed. The bolus of food is going down at the first part of the esophagus. And then the esophagus starts contracting behind the bolus of food, and it goes down through the esophagus to the stomach. And then the epiglottis reopens so you can breathe again. Then we deal with peristalsis and segmentation. Peristalsis and segmentation, they can occur together in an alternating fashion. So first, peristalsis, that's a wave-like ripple of the muscle layer of a hollow organ. So the esophagus is a hollow organ, and it can have a wave-like ripple of the muscle layer. The stomach has peristalsis. The small intestine, large intestine have wave-like ripples. Segmentation is a mixing movement. So digestive reflexes cause a forward and backward movement with a single segment of the GI tract. So peristalsis moves along the entire length of that hollow organ. Segmentation is mixing movement in single segments. We'll talk about that. Here's peristalsis. So when a bolus of food is going down, so let's say this esophagus, bolus of food is going down, where it stretches, the muscles relax to allow it to go forward, but behind it, it contracts. So if there's a stretch, then it'll start to contract. So as this moves from this contraction, this moves forward, then it contracts right behind it. So as this stretches, this muscle now will start to contract. And so it just moves along and it contracts and moving the contracts in a certain direction and moves the bolus going forward. Okay. And this just shows the pressure as you go through the esophagus. So this is the upper esophageal sphincter. That's the very beginning of the esophagus. Then you have lower esophageal sphincter down here. And it's just saying that when you have, after swallowing, this is the amount of pressure. And so you can see the amount of pressure goes up as you get further along, and then it gets it closer to the stomach, the pressure actually goes down as it's going into the stomach. So this is segmentation. Remember we said that with peristalsis, you have the bolus, you have contraction behind the bolus, and it loosens after it. With segmentation, you have one bolus, so this was one bolus, and in between the bolus, you have multiple areas of contraction. Okay, and it's like ring-like contraction, and it kind of breaks up that bolus. That bolus is fluid, typically fluid. It's a lot of fluid, and it can be broken up, from one bolus into multiple sections. And then as it goes down further, it comes through like one of these balls, but in between that ball, it starts breaking up once again. It keeps breaking it up segmentally until it gets really small pieces of food. Okay. Okay, now how is motility through the gastrointestinal tract regulated? Well, first you have gastric motility. So food in the stomach is churned, which has propulsion and retropulsion. Propulsion is pushing it forward, retropulsion is pushing it back. So that's how it churns. It gets pushed forward, then pushed back, pushed forward, mixed back. And then when it's doing that, it's mixed with gastric juices to form chyme. So that bolus mixes with gastric juices and it becomes, we can now call it chyme. So chyme is ejected about every 20 seconds into the duodenum. It just ejects a very little bit at a time. So an empty in the stomach takes approximately 2 to 6 hours, depending on how much is in there and what type of substance you're breaking down. So what you ate. Gastric emptying is controlled by hormonal and nervous mechanisms. So intestinal motility. So intestinal motility mixes chyme with digestive juices from the pancreas, liver, and intestinal mucosa. So the duodenum specifically, the first part of the small intestine, gets substances from the pancreas that gets deposited into the duodenum. Gets substances from the liver and in the intestinal mucosa itself, the lining of the mucosal wall. Then peristalsis rate picks up as the chyme approaches the end of the jejunum. Remember we talked about the different segments of the small intestine. You have the duodenum first, then you have jejunum, then you have ileum. And so it's moving it through the small intestine into the large intestine. So peristalsis rates, they pick up, it gets faster as the chyme gets to the end of the jejunum. And the reason for that is the duodenum is the main area where you can get absorption. That's where nutrients are going to go from the intestines and through the intestinal wall, through the cells of the intestinal wall, and into the bloodstream. After it moves through there, then it moves quicker. It doesn't have to absorb as much. It's starting to get ready for defecation. So after the chyme leaves the stomach, its passage all the way through the small intestine takes approximately 5 hours. So now we're getting the learning objectives for the next lesson, which is digestion, excretion, and absorption. So we're going to define chemical digestion, define the term enzyme, classify enzymes according to the type of chemical reaction catalyzed, list and discuss the important properties of digestive enzymes, and we're going to compare and contrast protein, fat, and carbohydrate digestion. We're also going to discuss salivary, gastric, pancreatic, biliary, and intestinal exocrine secretions and how they are controlled. We're going to identify and discuss the absorption of nutrients resulting from the digestive process and the structures into which they are absorbed. Then we're going to discuss elimination and defecation. So first, let's look at chemical digestion. Chemical digestion is the chemical changes that result from hydrolysis. Remember, we actually talked about hydrolysis. Hydrolysis is adding of water to break something up. So chemical digestion, the chemical changes that result from that hydrolysis. Now we're talking about catabolic reactions. And since we're talking about catabolic reactions, catabolic reactions are, if you want to break it down, it's endothermic or exothermic. Exothermic, very good. You're actually giving off energy. Digestive enzymes, you have extracellular organic protein catalysts. So the digestive enzymes that are used in chemical digestion are extracellular organic protein catalysts. So digestive enzymes, when you say it's extracellular, that means the enzymes that are used outside of individual cell. And they're organic, which they are protein, so the enzymes are protein, and they're catalysts. They help move along a reaction. So it means that the digestive enzymes, they operate in lumen of digestive tract. So they're outside of any body cells, but they're in the empty space in the digestive tract. So inside the stomach, inside the small intestine, large intestine, they're in the lumen, empty space inside those. Some properties of chemical digestion is they're very specific in their action. Certain enzymes only act on certain things, and they function optimally at a specific pH. So certain pHs, they will work best. Other pHs, they won't work. So properties of digestive enzymes. So most enzymes catalyze a chemical reaction in both directions. So an enzyme for a particular substance will break it down or help build it up. Enzymes are continually being destroyed or eliminated from the body and must continually be synthesized. So most digestive enzymes are synthesized as inactive proenzymes. And that's important to know that most enzymes are produced and they're not active. They're only active when the body wants them active. So they're produced as proenzymes, which are inactive enzymes. And then when they get activated, then they are active enzymes. So one of the ways that you can activate an enzyme is by the pH being a certain amount. And so, for example, with pepsin, pepsin is produced as pepsinogen. And so our normal pH in our bodies is about 7, just about 7. It's a little bit more basic, but it's around 7. Pepsin is produced as pepsinogen, and pepsinogen is activated when the pH becomes very acidic. And so when the pH becomes acidic, then it's pepsinogen is converted to pepsin. So you can see the importance. Pepsin is an enzyme that breaks down protein. So it is not active unless it is very low pH. So that means even if it got loose somewhere else in the body, it's not activated. The active form of pepsin is not activated until the pH is very low. So most of the other parts of the body, other than the stomach, does not have pH as low as 2 and 3. And so even if this enzyme got out in different parts of your body, we would not have to worry about it breaking down protein inappropriately because the pH would not be low enough. So then another one is trypsin. Starts off as trypsinogen as the proenzyme, and then it becomes trypsin when the optimal pH. Optimal pH is between 5 and 9, or 4 and 9. Now we have carbohydrate digestion. Carbohydrates are saccharide compounds. Polysaccharides are hydrolyzed by amylases to form disaccharides. So they're broken down, then they produce disaccharides. So the final steps of carbohydrate digestion are catalyzed by sucrase, lactase, and maltase. So catalyzed, that means that these are enzymes that will help get a reaction to occur. Okay, so and when you see ACE, that typically means it's an enzyme, and usually it is reference to whatever it's actually working on. Like sucrase is an enzyme that breaks down sucrose. Lactase is an enzyme that breaks down lactose. So what compound does maltase break down? Right, maltose. So maltase breaks down maltose. Lactase breaks down lactose. Sucrase breaks down sucrose. And these break down, they're found in the cell membrane of epithelial cells covering the villi that line the intestinal lumen. So we'll talk about that in a little bit. But on the villi, which is on the villi, are line the intestinal lumen of the duodenum. So it's the intestinal wall around the opening in the duodenum. And their villi that help increase the surface area. And these enzymes that aid in carbohydrate digestion, that's where they're found. So these are broken down into simpler sugars. And so ultimately you want to break them down to glucose. And then the glucose can be transported across the intestinal wall through the cells and get into the blood. So it breaks down to the simple sugars. So, and this with salivary amylase will start the breakdown process. So you take in a polysaccharide, and then at the very beginning, so this represents intestinal lumen. So your intestinal lumen begins in the oral cavity. So your mouth is a hollow tube that is tube that's going from your mouth all the way down to the anus. And so in this hollow tube, you take in a polysaccharide, salivary amylase is produced, which starts breaking down. And also you have pancreatic amylase that is deposited into the duodenum, starts breaking down polysaccharides into disaccharides. And then the disaccharides are broken down into the monosaccharides. And you see the enzymes that are utilized here are the sucrase, lactase, maltase. This is in the this hollow tube in this part is in the duodenum. You break down into the monosaccharides. And that's where you see some of these disaccharides. They actually are acted on by the enzyme sucrase, lactase, maltase, and break down to the monosaccharides. Monosaccharide being glucose. Very good. So now that was carbohydrate digestion. Now let's go into protein digestion. So protein compounds are made up of twisted chains of amino acids. Remember, amino acids are amino acids bound together by peptide bonds, and many peptide bonds together is polypeptide chain. And polypeptide chains put together make up a protein. So proteases are enzymes that break down protein. They catalyze hydrolysis of proteins into intermediate compounds and finally into the amino acids. So proteases break down proteins into not as complex proteins, so they're intermediate. And then finally, the proteases will break down these intermediate compounds into amino acids. So here are the main proteases. We have pepsin, that's found in gastric juice, trypsin in pancreatic juice, and then peptidases in the intestinal brush border. That's on the villi in the duodenum of the small intestine. On the villi has a couple thousand small little hair-like extensions that are called the intestinal brush border. So here with protein digestion, you take in a protein, so you eat a hamburger, you take in that protein, it starts breaking down. Actually, protein digestion does not occur until it gets into the stomach. So protein goes down and gets into the stomach, and then proteases, the proteases are in the gastric juices and also in the pancreatic juices. Pancreas secretes its enzymes into the duodenum. So that's where it starts to break down the proteins, and it breaks down into peptides. And then the intestinal peptidases break down the peptides into amino acids. And then the pancreas secretes its substances into the uh, duodenum of the small intestine, and the proteases that it produces, that are producing the pancreatic juices, break down into amino acids as well. So you have intestinal peptidases and uh, the proteases in pancreatic juices that all break down the protein into amino acids. Now then you have fat digestion. So fats must be emulsified by bile in the small intestine before being digested. So the pancreatic lipase is the main fat digesting enzyme. But before the fat can be digested, it must be emulsified. We'll talk about that in the next slide. So the residue of digestion, so the residue is what we did not absorb, so what our body does not want to use. So the residue of digestion, so some compounds of food resist digestion and are eliminated as feces, this waste product. Let's talk about this emulsification a little bit. This is the process of emulsification. You have a product called lecithin, and lecithin actually it starts off here. This is lecithin. Lecithin has a hydrophobic head, which is polar. I'm sorry, hydrophilic head, which is polar. And so if it's polar, hydrophilic means it likes water. Okay? And it has a hydrophobic tail. Hydrophobic means it doesn't like water. It's non-polar. So what happens is when you have oil separated from water, because water and oil don't mix. When you mix in lecithin, so you mix in lecithin, what happens is the lecithin traps the oil inside. The hydrophobic tails face the oil, and the hydrophilic head faces the water. So now you have packets of oil floating around inside the water. So in essence, it mixes, but it doesn't mix totally within the water. It's just that this package, this is how it looks. This is one of them here, and blow it up. This is the oil. This is the hydrophobic tail of the lecithin. These are the hydrophilic head of the lecithin. And so it carries this oil and is able to let it distribute within this water. And that's called in the cell. So this is what we call emulsifying the fat. So this is what happens. You get large fat droplets that go into your intestine, and then you have bile that's released from the liver. The liver continuously secretes bile, and that bile goes in, and the bile has the lecithin, which actually surrounds these large fat droplets and breaks it down into smaller fat droplets, and they form these. They're able to, when the water and the fat mixes and it separates into smaller portions, then you have pancreatic lipase, which actually will break down fat, digest fat when it's in the smaller components. Then the pancreatic lipase is able to work and act on the fat droplets. So the fat needs to be emulsified first by the bile from the liver, and then the pancreatic lipase can work on it to actually digest the fat. I need you to understand that the bile emulsifies the fat, large fat particles, breaks it down into smaller parts, and allows it to mix with the fluid in the digestive tract. And then the pancreatic lipase is able to digest the fat. Without emulsification from the bile, then the pancreatic lipase can't work on it, doesn't work on it effectively. So in this picture also, I want to just describe that the liver continuously produces bile, and when the body doesn't need bile, when it doesn't need it, it's not digesting fat, then that bile is stored in the in the gallbladder. That's the gallbladder here. So if someone has cholecystitis, which is inflammation of the gallbladder, and has a cholecystectomy, which is removal of the gallbladder, so you just cut the gallbladder out because of gallstones or whatever, then the person still will produce bile, but it's like they won't have a storage area for bile. So if you have a lot of fat coming in at one time, the constant production of the bile will not be able to break it down effectively, and so their stools will have fat in it. But they will absorb, enough fat to, for survival. So you don't need the gallbladder, but it's just effective when you take in a lot of fat. Then the stored and concentrated bile in the gallbladder can be released and emulsify the fat so that the pancreatic lipase can digest it. Okay, now let's talk about secretion. You have different secretions. You have saliva, which is secreted by salivary glands. Then you have mucus that's secreted, which actually lubricates food, and with water facilitates mixing. It's easier to mix when it's fluid. Then you have amylase, which is secretion. That's an enzyme that begins the digestion of starches. So you have secretions from the salivary glands that will actually secrete amylase. So a small amount of salivary lipase is released, and its function is uncertain. But the amylase that is released in the salivary secretions starts the digestion of starches. Then you have gastric juices. The gastric juices, that's the juices that are in your stomach. So the gastric juices are secreted by gastric glands. So a major one is pepsin. So pepsin is a protease that begins the digestion of proteins. And the inactive form or the proenzyme of pepsin is, right, pepsinogen that we talked about before. Now, the hydrochloric acid in stomach is secreted by parietal cells. That contributes to the gastric juice. So hydrochloric acid decreases the pH of chyme for activation and optimum function of pepsinogen. The HCl actually activates the pepsinogen, cleaves off different portions of it to form pepsin. So the gastric juice is released actively into the gastric juice. I'm sorry, the HCl is released actively into the gastric juice by the hydrogen potassium ion pump, ATPase pump, also called the proton pumps. So I'm going to repeat that. The hydrochloric acid, which is secreted by parietal cells, that hydrochloric acid is released actively into the gastric juice by the hydrogen potassium pumps, proton pumps. This is kind of how it works. This here is the hydrogen potassium pump. It actually pumps hydrogen ion into the gastric lumen, and it pumps out potassium. And so inside the gastric parietal cell, you have H2CO3, which is, you have CO2, H2O, form H2CO3. It releases hydrogen ion, and it releases bicarb, and the hydrogen ions pumped into the gastric lumen, potassium is pumped out. And see, the parietal cell is in direct communication with the blood vessels. And that's where you can get rid of the bicarb that is produced from the CO2 and H2O. And also the chloride comes from the blood as well, so that the chloride can be in the gastric lumen and combine with the hydrogen to form HCl. Now the chloride is pretty much always kind of around. It's the presence or absence of the hydrogen ion that will determine the pH. It will determine if it combines with the chloride to form the HCl. Okay. All right, and the ATP here just says that this sodium potassium pump is pumping against its concentration gradients, against the hydrogen ion concentration gradient and against the potassium concentration gradient. So you need a form of energy to pump against the concentration gradient. And so that energy comes in the form of ATP. Then this is just the cell membrane surface of the parietal cells. And it just shows in terms of the different vesicles that release the substances and the microvilli that are on the surface that increase the surface area. So gastric juices, they release intrinsic factor, which is secreted by parietal cells. The intrinsic factor protects vitamin B12 and later facilitates its absorption. So intrinsic factor, it actually facilitates or helps the absorption of vitamin B12. And so what that says, if your stomach lining is not working properly, it's not releasing intrinsic factor, then you can be deficient in vitamin B12. Vitamin B12 is very important for a number of processes in the body. So that's intrinsic factor. Actually, I want you to know that in terms of intrinsic factor. Intrinsic factor is necessary for protecting and absorbing vitamin B12. And then also the gastric juice has mucus and water that lubricates, protects, and facilitates the mixing of chyme. All right. Now we have pancreatic juice. Pancreatic juice is secreted by acinar and duct cells of the pancreas. These are the cells that are involved in the exocrine function of pancreatic tissue. So proteases such as trypsin and chymotrypsin are secreted by the acinar and duct cells of the pancreas, and they are enzymes that digest proteins and polypeptides. Then also we have lipases. Lipases are enzymes that digest emulsified fats. That's what we're talking about. Fats that, the enzymes that digest fat, and more specifically, they digest emulsified fat. That's what we're saying, that fats will not be digested by the lipase or the pancreas until they're emulsified by the bile. Then it also secretes nucleases. Nucleases are enzymes that digest nucleic acids, such as DNA and RNA. Then you have amylase. Amylase is an enzyme that digests starches. Where did we see the name amylase before? Right, a couple slides back, it said in the saliva, some of the salivary glands will secrete amylase to digest starches. Okay. Then talks about the bicarbonate secretion. This is just saying in terms of the pancreatic duct, and as it's developing its bicarb to actually, bicarb is produced to actually be sent to the duodenum. Bicarb is basic, and so the pancreatic ducts actually deposit basic substances into the lumen of the duodenum to offset the acidic nature of the chyme. Okay, this is kind of a description of the pH balance related to the digestive tract. Whoops. Okay, so what it's trying to say here is that with the digestive tract, it's in direct communication with the blood, and so you have uh, the hydrogen ions, and the pancreatic cell sends in bicarb, and the hydrogen ions that have been sent in with the parietal cell because to form the HCl, and the bicarb that's brought in from the pancreatic cells will absorb the hydrogen ions, form the H2CO plus, and this will be sent out as CO2 and H2O. That's how the bicarb that's produced by the pancreas will help to increase the pH in the duodenum. So the hydrogen ions that are free floating from the acidic chyme, the bicarb that is sent in from the pancreatic duct cells, they combine, and then you'll have CO2 and H2O, which are not acidic. And then, so as it goes down the digestive tract, then it becomes more basic. Okay, then we have bile. Bile is secreted by the liver, is stored and concentrated in the gallbladder. So the lecithin and bile salts in bile emulsify fats by encasing them in shells to form tiny spheres called micelles. That's what we talked about earlier. Sodium bicarb increases the pH for enzyme function. So there's bicarbonate in the bile as well. And so the bile is secreted from the liver into the duodenum. Remember, the duodenum is receiving chyme from the stomach, and that chyme is acidic. So you have pancreas sends in sodium bicarb, and also the bile in the from the liver secretes sodium bicarb as well, so that the pH can be raised, and so you can have optimum enzyme function. Then the bile is also associated with cholesterol regulation. So cholesterol is a product of detoxification, and then bile pigments, such as bilirubin, are waste products excreted by the liver and eventually eliminated in the feces. And so this is normal process in terms of how the body, and the only way the body actually gets rid of cholesterol. Now you have intestinal juices. This is an intestinal wall directly. It's intestinal juices are secreted by the intestinal exocrine cells. Remember what we say, exocrine cells? What represents exocrine cells? How can you describe exocrine cells? Right. They're glands or cells that actually have ducts associated with them. So whatever it's producing, it's actually sending it through a duct directly to where it wants to act. So in this situation, it's secreted directly into the lumen of the duodenum. And so you have the secretions, the intestinal juice that's secreted by the exocrine cells include mucus and water to help to lubricate and aid in the continued mixing of chyme. Then it also secretes sodium bicarb, increasing the pH, once again for optimum enzyme function. Now, the salivary secretion. Salivary secretion, the saliva is secreted, its only reflex mechanisms control the secretion of saliva. So you only have reflex mechanisms that control the secretion of saliva. So chemical and mechanical stimuli, so some of the reflex mechanisms can be chemical or mechanical stimuli. They come from the presence of food in the mouth. So the mechanical stimulation, you can see physically as you have something that physically contacts your mouth, and fills your mouth, then it will stimulate saliva production. And then also chemical. When you actually have something that's starting to get digested and your body recognize it as a chemical that you want to absorb or to digest, then it will produce saliva. Then also you have olfactory, which is nasal, and visual stimuli. So olfactory, so what you smell and what you see come from the smell inside of food. So that will stimulate salivary secretion. You know that all too well. If you walk into your favorite aunt's house or grandma's house that is cooking Thanksgiving dinner and you come in hungry and you smell that food, first thing in your mouth starts watering. Okay? Or if you see a good meal that you're like, oh, that looks so good, your mouth literally starts watering. And that's the reflex mechanism for secretion of saliva. Now, the control of gastric secretion. Actually, I want you to know these different phases. So definitely know these different phases. You have cephalic phase. Cephalic usually makes reference to your brain. So it's called the psychic phase because mental factors activate the mechanism. So the vagus nerve stimulates production of gastrin. Gastrin is produced by G cells in the stomach. Okay, so know that gastrin is produced by G cells in the stomach. And the cephalic phase of gastric secretion is the psychic phase. Once, like I talked to you about your grandma or your favorite aunt's Thanksgiving dinner, and you think about it, and the years passed, and you ate, and you can start the secretion process. So that digestive process in your stomach will start to churn and start generating enzymes just with the thought of that food. So when you're thinking about a meal, and when you smell it, when you see it, it starts everything churning and you start feeling hungry and everything's getting ready. That's the psychic phase or the cephalic phase. Then you have what you call the gastric phase. So gastric phase is when the products of protein digestion reach the pyloric portion of the stomach. Then they produce the release of gastrin. So once again, gastrin is produced literally when you take food in and the products of the protein digestion. So when you chew for the mechanical digestion, and then when the chemical digestion in the stomach, and you have that churning of the chyme back and forth um, when the products are actually touch the part of the stomach in the pyloric region at the very end of the stomach. When it reaches that part, then that stimulates release of gastrin. So then you have the intestinal phase. So various mechanisms seem to adjust gastric secretion as chyme passes to and through the intestinal tract. So gastrin is released and produced in 3 different phases. Just when you think about it, or you smell it, or you see it, then gastrin's produced. When you physically have protein broken down to a certain extent and it goes into the pylorus region of the stomach, more gastrin's produced. And then also as the chyme passes to and through the intestinal tract. So if you get to a place where chyme gets to and getting through the intestinal tract, then more gastrin's produced as well. Okay, and then we have control of pancreatic secretion. Remember, pancreatic secretion occurs, it's secreted into the duodenum. So pancreatic secretion is stimulated by several hormones released by intestinal mucosa. So the lining of the intestinal wall itself will secrete several hormones that will stimulate pancreas secretion. So secretin evokes the production of pancreatic fluid, low in enzyme content but high in bicarbonate. So if you think about that, secretin from the intestinal wall, secretin is stimulated because of high pH. And so with high pH in the intestinal wall, from the chyme that comes into the duodenal intestinal wall, it will send signals to the pancreas and tell the pancreas, I need you to produce bicarbonate. Bicarbonate is very basic. It will help offset the acidic nature of chyme. So it will tell the pancreas to actually secrete a lot of bicarbonate to help neutralize this acid. So that fluid that the secretin uh, stimulates from the pancreas is low in enzyme, but high in bicarbonate content. Then also you have CCK. So CCK is cholecystokinin, and so it causes cholecystokinin causes increased exocrine secretion from the pancreas. So this cholecystokinin will cause increased exocrine secretion from the pancreas. So the exocrine secretion, when you see exocrine secretion, you immediately think of what? What is exocrine? Right. It means that it has a duct. So exocrine secretion from the pancreas means that it's secretions that's going to go directly from the pancreas, go through a duct, and empty into the duodenum. So CCK opposes gastrin. So it inhibits gastric hydrochloric acid secretion, where gastrin was stimulated when you saw food or when you had the breakdown of protein or when that chyme was passing through the duodenum. And gastrin stimulates hydrochloric acid secretion. It stimulates everything in terms of breaking stuff down. Now the CCK goes against gastrin. So when CCK is produced, now food has already gotten into the duodenum. Now CCK says, let's offset the gastrin, turn off the gastrin, turn off the hydrochloric acid. We need to neutralize the acid, and we need to get to the process of absorption. So it opposes gastrin, inhibits gastric hydrochloric acid secretion, and it stimulates contraction of the gallbladder. So that bile is ejected into the duodenum. So CCK causes bile to be the stored bile and concentrated bile to be secreted into the duodenum, so that if there was food in there that was fat, large fat particles, that can be broken down as well. So now the control of bile secretion and intestinal secretion. So bile is secreted continually by the liver. So the intestinal secretion, well, little is known about how intestinal secretion is regulated. Some suggest that the intestinal mucosa is stimulated to release hormones that increase the production of intestinal juice, but we're not quite sure. But just know that in terms of the control of bile secretion and specifically intestinal secretion, that bile secretion is secreted continually, and intestinal secretion we really don't know much about what regulates the intestinal secretions. Then we go through absorption. Absorption, the process is passage of substances through the intestinal mucosa into the blood or into the lymph. And the lymphatic capillary around the intestine are called what? What are them in lymphatic capillaries that are around the intestine? What are they called? They have a specific name. Right. The lymphatic capillaries that are around the intestine are called lacteals. So lacteal lymphatic capillaries. So the mechanisms of absorption that Other substances are absorbed through more complex mechanisms, but the secondary active transport, that's how sodium is transported, sodium co-transport, you don't have to worry about that. But mechanisms, um, what we refer for absorption, the main ones that are absorbed are the glucose, the peptides, the fatty acids. They are broken down to the basic components, and those are passed through the intestinal mucosa into the blood or the lymph. These other ones that they're talking about, they talk about more complex, and I'm not going to ask you about that specifically. But the absorption I want you to be concerned with are the ones that are the basic components: glucose, amino acids, fatty acids. So an intestinal wall is made up of villi. And so the intestinal wall, the villi, each one of there's a whole lot of villi here. And actually, this is one villi here, and then you have uh, the capillaries, the arterial and venual, and the inside. This is kind of a schematic of it. On one side, you have the arterial on one side, and on the other side, you have the venual and capillary bed in between. This thing that's in the middle is the lacteal, which is the lymphatic capillary. So every villus has one arterial, one venual, and one lacteal. Okay? And then on the edge of the villi are microvilli. This is this brush border, and it's on each of the cells of the villi, and it's just a lot of little small hair-like uh, structures that increase the surface membrane for absorption. So on here, as it says, this is the lumen of the duodenum. And so water will go in to the bloodstream, sodium goes in, glucose goes in, amino acids go in, fats go in. But you see, fats actually go into the lacteal. Fats are actually too big to go into the blood vessel walls, and so fat goes into the lacteal. But all of these are transported into this extracellular space that's around this capillary bed, but it's just that fats can't go into the blood vessel, so fats go into the lacteal. So absorption, you have transcellular. Transcellular is across cells. Transcellular absorption moves nutrient particles through cells. And paracellular absorption moves particles between cells. So it's saying transcellular, it goes from one cell through a cell, and it takes it through the cell into the bloodstream. Paracellular is where it goes from cell to cell. Nutrients that are absorbed into the blood, such as monosaccharides and amino acids, monosaccharides being glucose, first travel to the liver via the portal system. So all that's saying is when you absorb the nutrients into the blood, then you have the carotid portal vein that instead of going directly to the heart through the vena cava, it actually goes to the liver. And then the liver is able to detoxify, able to take out any of the excess substances, able to process it before it sends it on to the heart. Then we have elimination. With elimination is the expulsion of feces from the digestive tract. This is referred to as defecation. So defecation results from a reflex elicited by stimulation of receptors in the rectal mucosa. The stimulation is caused by distension of the rectum. So when the rectum gets distended, then you get an impulse to actually defecate. Constipation is the contents of the lower part of the colon and rectum move at a slower than normal rate. Then you have diarrhea. Diarrhea results of increased motility of the small intestine, which causes decreased absorption of water and electrolytes and a watery stool. All right. Any questions? All right. Remember, on this particular quiz that we're going to have for this chapter, this quiz is not going to be until uh, Thursday. So beginning of class Thursday, 5 o'clock, we'll have a quiz on this chapter. All right, so do well. I'll talk to you soon.